you can hear me? Perfect. Okay, good. Yeah, my presentation will actually in some way continue Pascal's presentation and also pick up some of the points we were discussing yesterday. How can we distill real understanding about mechanisms from the big data? So how can we go beyond just making good predictions and also linking genotype to phenotype, this big question. So the question that I will address here is, what determines whether a genetic variant has an effect in the molecular network that is confined to a small part of the network, as opposed to affecting large parts of the network, having these long-range effects in molecular networks. So what determines whether effects are local or global? I will also talk very briefly about a paper that we just published. Because it's published, this part will be very brief, so you can read the paper. Okay, so what we want to know is, how do genetic variants impact cellular traits? And for this, we are looking at molecular networks because they actually mediate these effects. So we do eQTL, protein QTL, and other QTL studies. Most people are looking at concentrations of molecules, concentrations of transcripts, proteins, and small molecules. How are they affected by genetic variants? And then they do the QTL studies. We've just published a work where we are looking at states of proteins. And I think this is really the next level. We have to go beyond concentrations. We also have to look at states of molecules. So in this published paper, we have done the very first phospho-QTL study, phospho-proteome QTL study. So this is the paper. It came out, I think, like 8 weeks ago or something. And just briefly, the study, because the data is actually relevant also for what I'm going to show afterwards, the unpublished work. So this is based on a cross of 2 yeast strains, RM and BY. We didn't do this. This was done by Leonid Krugliak and Rachel Brem many years ago. So this is a very well characterized population of genetically heterogeneous yeast strains. And this is great because there's a lot of background knowledge that we can utilize for the interpretation of the data. So we have this cross. And then we took cultures from these cells and subjected them to RNA sequencing, protein quantification, so swath proteomics or DRA proteomics, and then the phospho data. So this was the innovative part. And I think it was really important that we had all 3 layers, omics layers from identical cultures, which really increases the ability to compare the data between these different layers. Important to know is that the protein concentrations will be correlated with the mRNA concentrations. So this is what you see here. So there's a lot of positive correlation between proteins and their encoding RNAs, the encoding mRNAs. But there's also correlation between phosphopeptides and the proteins of which they are part. So this kind of it's trivial, right? If mRNA goes up, the protein will also go up. So we wanted to distinguish effects that are mediated through the mRNA at the protein level from effects that are directly targeting the protein, for instance, through modulating translation of the protein or protein stability. So those effects would be independent of the mRNA. So these are things we want to distinguish. And the same is true for the phospho effects, because if a protein is upregulated, then the phosphopeptide or the residue of the phosphorylated amino acid will also go up. That's kind of trivial. So we want to distinguish effects of, for instance, kinases modulating phosphorylation of the target protein from effects that are just due to abundance changes of the protein. So the way how we did this was relatively simple. We just did regression. So we regressed the phospho level of the level of a phosphopeptide as a function of its protein, of which is part. So here each dot would be 1 strain, and we're always looking at the same phosphopeptide across these different strains. And then we're doing a linear regression, and then we're saying this is the variance of the phosphopeptide that is explained by protein level variation. And then we regress out this effect in order to focus on effects that are independent of the protein abundance. So we're looking at the residuals. The residuals are noise to some extent, and to the other extent, it's a variation in phosphorylation that is independent of protein abundance. We did the same with the protein-RNA relationship. So we again regress protein levels as function of their mRNAs, and then we do the same trick. So we say that this is the variance that is explained by the mRNA, and the remaining variation, the residuals, are due to post-translational effects or post-transcriptional effects or noise, of course. So we create new traits. So now we have the mRNA level as a trait that we can map. We have the protein level as a trait we can map. But we can also map these residuals as new molecular traits that are derived from the other molecular omics layers. So we can now ask which genetic variants are affecting these residuals. So this is what we did. We did this regression, and now we have 5 omics layers that we can map as molecular traits to the genome. We have the mRNA, we have the protein abundance, we have those residuals as an intermediate layer, we have the phosphopeptides, and again, the residuals as an intermediate layer. The way how we do a QTL mapping is using machine learning. So we like to use random forest. This has advantages compared to the linear methods, which are more frequently used because random forest accounts for nonlinear effects, it accounts for interactions between different loci. So we've shown in the past that it actually is more powerful than linear QTL mapping methods. We have many papers on this. We like it because it's very intuitive, it's very robust, so there's no tuning that you have to do. You can easily parallelize it because it's based on training trees, so each tree can be trained on one node if you want. It accounts, as I said, for the static effects, and you can also explain it to an audience without showing any equations, which is not relevant in this audience, but in other audiences, it does matter. Okay, so here are the papers that we've published on this, if you're interested in this approach. So now some data. I have only 2 slides on this because, as I said, it's published. So first is how are genetic effects that change mRNA levels mediated to the protein level. And essentially, it's a one-to-one relationship. And this is not trivial. So what you see here is the allelic effect on protein levels and then the allelic effect on the encoding mRNAs. And if you regress through all proteins, you get almost a slope of 1. So this is a perfect correlation, not a perfect correlation, but it's a bit like a perfect transmission of the effects from mRNAs to proteins. And that disagrees with earlier work. And I think this is mostly because earlier studies didn't have the same sensitivity at the proteome level. So they were biased for high abundance proteins, which are mostly ribosomal proteins. And because now the technologies are much more sensitive, we have a less biased view of the proteome. And I think now we see the truth, which is that most of the effects are directly transmitted to the protein level. So these are the ones in gray. But we do have some exceptions. We have the green proteins here. This is very interesting. These are in fact effects that are enhanced at the protein layer. So they are stronger than expected based on the mRNA. And then we have the purple ones, which are buffered effects. So these buffered effects where the effect on the protein is weaker than on the mRNA is mostly due to stoichiometric control. So if you overexpress a protein, but the protein is not included, for instance, in a ribosome, if it's a ribosomal protein, it will be degraded. So that is buffering this overexpression effect. The second, one second important finding, there are more, is that the phospho layer really helps in interpretation of cellular traits. So we've looked at hundreds and hundreds of different cellular traits, compound resistance and morphological traits. And it turns out that the phospho levels explain the variation of those traits much better than protein levels, the green is protein levels, or mRNA, the blue. So it really is important to look at protein states and especially the signaling. This is what the phospho layer really represents, the signaling that's going on in the cell. Okay, now the new work. So now we want to map network states. So essentially, we want to view the state of a molecular network as a trait that you can map genetically. So that you can say, okay, this variant is modulating this network state. In particular, we're interested in the question of the difference between long-range effects and local effects. So long-range effect would be an effect that is modulating large parts of a network and then eventually changing some cellular trait. And those are mostly also pleiotropic effects. So this is the link to Pascal's presentation. While local effects are usually much more specific, they only affect a small number of traits, if maybe just one. So what distinguishes these two types of effects? So the The question is, what are genetic effects propagating through large parts of the molecular network, as opposed to when they are confined to small parts of the network, so local versus global. My conception of this is that if you look at this very high dimensional space that is created by the molecular traits that we can quantify, so all the transcript levels, all the protein levels, this would create a 20,000 dimensional space. In yeast, not that many. Then many combinations of traits in this space are inviable, so they're not possible. So the cell would just die if you would have a specific combination. So this means that only a very small subspace of all possible trait combinations creates a viable cell. And then out of those, most are actually in favored through evolution because they are not fit. They are viable, but they're not fit. So there's selection against those combinations of traits. So that further reduces the space of, let's say, observable combinations of molecular traits that you find when you're looking at natural genetic variation. So if you want, you could say that this is kind of a manifold, in this high dimensional space, and the network is essentially moving on this manifold when you have genetic variation. I think that if you do knockouts, you could easily push the cell out of this manifold. So this is a problem with knockouts, that sometimes they create a state that is to which the cell is not adapted, and then you get a non-physiological response. But here we're looking at natural genetic variation, so most of the allele combinations happen in nature as well, so there's at least no strong selection against them. So this is the space on which the network is moving. So if this is our space, then the green dots would be observed states we have in our cross, in this particular cross. And then the yellow ones would be states that you may observe if you created new allele combinations. That's the idea here. So this is an important notion, because that means that if you change some part of the network, other parts of the network will have to adapt. So you have to respond. These are the long-range responses of the network. So for instance, a very well-known example in yeast is that if you change glycolysis, if you downregulate glycolysis, oxidative phosphorylation will go up. And if you reduce oxidative phosphorylation, glycolysis will go up. So there's this balance between these two processes. And so what I'm saying here is that there are different levels of stoichiometry in the cell. We have stoichiometric control at the level of protein complexes. This was what I was showing before. If you have a protein change, it's buffered. Yeah? You have stoichiometric constraints within one module. So if you have a specific metabolic pathway, this is where the term actually comes from, stoichiometry. And then what we are saying is that you also have stoichiometry at a much higher level between those modules. So this would be the example, we have two modules, one module controlling oxidative phosphorylation, another one doing glycolysis, and they're also coupled. And the claim here is that there are many, many more processes in the cell, or modules, whatever you want to call them, that are all coupled to each other. So this is what I'm trying to sell. We already have an example of this published 2 years ago together with Beyer on pyruvate kinase 1 in fission yeast. There's a mutation or variant in the lab strain that was used in the community for many, many years, and nobody noticed that this variant is actually changing the efficiency of oxidative phosphorylation. And so these cells were always turning on glycolysis to a small extent, while if you had other wild-type strains, they would only do, or much more do, oxidative phosphorylation. And then this is affecting many other cellular traits, like also growth and stress resistance, which will be important. So this is an example of these modules and the connections between them. So this is what my postdoc was doing, Matthias White. Everything that I'm showing now is from Matthias. He was interested in better understanding a complex trait, and he chose heat stress. So he did heat stress experiments on this collection of yeast strains, and then he quantified a trait, which is what he calls heat-induced lag, which is just the lag time that the cells need in order to recover from the heat stress. So it's a non-lethal, low-level stress. The cells survive, and then they recover, but they recover at different speed. And that's the trait. So the variation here, this is between the two parental strains. This is what he's quantifying as his trait. And now we want to know what are the molecular determinants of this variation of this complex trait. So first, you can do just traditional QTL mapping. This is relatively boring, but straightforward. So he identified 3 QTLs, 1, 2, 3, that all significantly correlate with the trait. So here on the y-axis, you have the trait, and then here you have the 2 alleles, either the BY allele or the RM allele. And you can notice that for 2 of the QTLs, if you have the RM allele, the lag time is larger. And on the 3rd QTL, it's reversed. So now the lag time is shorter. So there's some kind of balancing of alleles in the parental strains to keep in both parental strains this stress response at some physiologically favorable level. This is also called transgressive segregation. Now, at the next step, he wanted to understand what are really the molecular connections between the loci here and the high-level trait here. So what is connecting this? So he looked at protein levels. This is the same data that I showed before, which is already published. And he was just asking which proteins explain the cellular trait. Again, using random forest in this case. I guess a linear model would also do. It's not so important. But anyway, so these are the proteins that significantly explain the cellular trait. And these proteins, so this is the correlation with the cellular trait, but these proteins are also affected by genetic variants. And especially they are affected by these 3 QTLs. So here we have the 3 QTL 1, 2, and 3. Whenever you have an asterisk, there's a significant linkage. So this is a significant QTL. But you can see here as well, but you can see that there's a very consistent response, which is not significant on the level of individual proteins. But if you look at the set of proteins, you can clearly see that there's a response. Now we have 2 types of QTLs, which is very interesting. So we have one, this is what I would call a local effect. So here, this is a locus containing HSP104. This is a cis QTL that is exclusively affecting this one protein. And because it's a heat shock protein, it's obviously related to the heat stress trait. So this makes a lot of sense. So this is a local effect. It's not affecting anything else. And then you have these long range or global effects that are affecting large parts of the molecular network. And especially, this was very interesting, this cluster of proteins here, the red cluster, it's marked in red. Because all these proteins show a consistent response. So here, if you have the RM allele, they are upregulated. And here, if you have the RM allele, they are downregulated. So this again correlates with, these balancing effects. So then Matthias started to look into these proteins, asking what they are doing. And it turns out that they are all involved in PKA and TOR signaling, or that they're targets of PKA TOR signaling, and that they are targeted by well-known locus, which contains the RA2 protein. So this is a well-studied locus in this cross. And it's already known that this variant here at the RA2 locus is affecting a very large diversity of many cellular traits. So basically what we're looking at here is the PKA and TOR signaling in yeast, which is converting on a common set of transcription factors and genes, which are then controlled by these 2 pathways. I'm also using the P word, even though I don't like it. But anyways, so one thing is these pathways are also controlling the balance between oxidative phosphorylation and fermentation or glycolysis. So again, the story comes back. So then Matthias wanted to quantify the activity of these pathways. Because they converge, we can't really distinguish, when you look at the levels of proteins, you can't really distinguish the activities of these 2 pathways. So what he developed is an integrated score that is quantifying the combined activity of these signaling pathways, so PKA and TOR. And he did this by using direct perturbations of these 2 molecules, PKA, and so this is a PKA inhibition, this is TOR inhibition data, and then he identified proteins, these are the marker proteins in green and in blue, that are strongly associated with the activities of these 2 pathways. So this is the effect of this IRA2 locus on these proteins, and this is the effect when you inhibit PKA or when you inhibit TOR, and you can see, I hope it's quite easy to see that there's a very strong correlation, which is just saying nothing else but that IRA2 controls, this pathway. So then he was picking these colored proteins as markers so that he could in the future quantify the activity of PKA and TOR signaling in any cell, in any strain, in any condition. So now we have a PT score, PKA and TOR score. This is the PT score, which I will use in the remaining presentation. And this is, I think, the most important slide of my presentation, because the amazing observation is that this PT score, it's one value, a one-dimensional value. This one value correlates very strongly with large parts of the molecular variation in this cross. So here we have a PCA of the transcriptome data, a PCA of the proteome data, PCA of the phospho-proteome, and this is a PCA of the residuals when you regress out the protein abundance effect. And in all four cases, you have a very strong correlation between the PT score and the positioning of the strains in these PCAs. So each dot is one strain, and then it's positioned based on the respective molecular information. And the coloring reflects the PT score. And here you see, the first PC, the first principal component, very strongly correlates with the PT score. Here also, first component and here first component correlate very strongly with the PT score. And here it's the second component that very strongly correlates with the PT score. So that means that, a major part of the variation that we have can be projected, if you want, or reduced down to this one single value. So this essentially characterizes the state of the network. This was budding yeast. We also have new data in fission yeast. This is unpublished data from fission yeast in collaboration with Jörg Beyer. And we see the same thing. We have different conditions, unstressed and oxidative stress. And again, we see a very strong correlation with the PT score in stress and non-stress conditions. And maybe it's not unexpected, under the stress condition, the correlation is even stronger here. Coming back to the heat stress, we also have a very strong correlation between the heat stress on this axis and the PT score on this axis. And this correlation here is stronger than using any of the QTLs, including the ARA2 locus, which was the strongest QTL effect. So that shows that this PT score really integrates the effects of many different loci, and therefore reflects also, an integration of this complex genetic interactions. Here the marked strains are the parents. The parented strains, they don't, this is this buffering, they're always in the center.
Yes.
Yeah.
For me, it looks like it's setting only an upper bound, right? It's because, in the bottom left corner, the bottom right corner, you have very high PT scores and still a short lag, whereas the upper right corner is completely blank. So there's a correlation, but it's kind of
yes, but I don't know to what extent that's just noise or real biology. There still is some variation here along this axis. And this may be just an outlier strain, but it might also be real biology. Yeah, I don't know. We have to look into this.
Use as a confounder and condition on that in order to get the genetic variation that analyze that you're still here.
So regressing out the PT effect.
Yes.
Yes, I think we did not do that. But what we did is, so I'm not showing that, but what we did do is we regress out the ARA2 effect, for instance, and then we still have strong effects from the PT score, which is showing that it's really affected by more than just one locus. So it's really a complex trait. That's data that I'm not showing because of time reasons. Okay, so the take-home message here is, so one was the molecular state is majorly determined by the PT score. The heat-induced lag time is also determined by the PT score. And now we come to the take-home message, which is this balancing between different modules that's going on in the cell. So explaining really these long-range effects. So here each circle is a Go term, or if you wish, a module in the cell, a functional module. And on this axis, you have how much does this module correlate, how much do the protein levels, here's proteome, protein levels of this module correlate with the PT score. So you can have positive, here 0, you can have positive correlation or negative correlation with the PT score. And this is the y-axis is basically the significance. So here on the right-hand side, positive correlation with strong TOR signaling, PKA signaling is proliferation. Cells are dividing, they're growing like crazy. So here you have all the terms that are related to translation. And on the left-hand side, when the signaling is repressed, you have all the stress responses that are going on here. So here you have oxidative stress and also the heat stress. Yeah? So this explains the heat stress response. But you also, I hope you can see that there are many, many cellular functions that are correlated with the PT score. So this is this global rearrangement of the cellular metabolism that's going on when you fiddle around with the PKA and TOR signaling. And they're all connected. So now the claim that we have is that when you change one of those modules here through some genetic variant, that will have indirect effects on the other modules because they have to respond. This is really what's going on here, and that explains these long-range effects. These are just the other omics layers. Here we have the phospho and the phospho residuals, and this is very interesting because we see now new terms, especially when you look at the phospho residuals. Here on the right-hand side, we see, for instance, cell cycle, which is majorly regulated through phosphorylation signaling much more than abundance changes. So this makes a lot of sense. But here we now also see chromatin organization, histone modification, so epigenetic or modules which are related to epigenetic control. So it's a very wide range of cellular functions that are affected by PT signaling. So this is the model that we now have. We have local effects. Local effects are genetic variants that change only one protein or a very small set of proteins. Then we have regional effects where you have a pathway or a module. So maybe this is some regulator, a kinase, or a transcription factor, some signaling molecule, and then all the molecules that are dependent on the signaling molecule would change. So this would be a regional effect. But if this change would change the activity of an important cellular module, that would have long-range effects. So these are the global effects which we have here. So you change one module here, let's say glycolysis, and then oxidative phosphorylation will respond. So then you have these more long-range effects. And this is very consistent, I think, with Pascal, what you were just showing, that there's a lot of crosstalk going on between these modules. They're not isolated. Just to nail the story, now there's some network analysis and the last data slide. Here we look at different hotspots. So hotspots are loci in the yeast genome, which are affecting large numbers of molecular traits in this protein concentration in this case. And then we correlate the effect with the change of the PT signaling. So here's the ARA2 locus, and you have a very strong correlation with the PT signaling in this case, as we already know. So this is a locus that is heavily changing the PKA and TOR signaling. BOL2 has a negative correlation. And then we also looked at a third example, which is this locus containing ZAP1, where we have a very weak or even no change in PT signaling to distinguish different types of responses. And this would be a local or regional effect, sorry, not a local, but a regional effect. So it's still a hotspot, it's affecting many molecules, but this has much fewer effects on other modules. And to show this in a quantitative way, we did some network analysis. So we looked at the distances of these target genes in the network. So how closely are they connected in the target network? And this is the average distance, or the pairwise distances of the target molecules for these three loci. And then I hope you appreciate that for ZAP1, we have relatively short distances here. This is 2 and 3. So they have short distances, while the other two loci have longer distances. So you have a lot lower fraction of target genes here, but you have many more targets with long distances. So here, I don't know if you can see it in the back, but there's still dots here. So up to a distance of 8, which doesn't exist for ZAP1. So that means that the ZAP1 targets are much more compact in the network than targets of ARA2 and BOL2. Okay, so long-range network effects are mediated via modulating the balance between cellular processes. The PKA-TOR signaling majorly explains variation of molecular traits, at least in this cross, but we also have data, which again, I couldn't show because of time, in fish and yeast that confirms, I mean, I was showing one slide, but we have a lot more data. And this was amazing that this signaling, this network state can be reduced to a single score, to just a scalar. And I think this has very strong implications for animals. I think you all know the Warburg effect, which is also changing this balance between glycolysis and oxidative phosphorylation. This is also very important during development. So when cells go from an ES cell state to later differentiation states, the balance between glycolysis and oxidative phosphorylation is changing as well. But this also has major impacts on the general organization of the cells during differentiation. Obviously, this has impact on stress resistance. So the importance of TOR signaling is well known also in animals. I'm involved in many studies looking at aging. And for instance, suppression of mTOR signaling, insulin signaling is very well known to extend lifespan. And we have now also evidence that there are genetic variants in the human population which target these exactly same pathways, the autologous pathways in humans, and also extend lifespan in humans. Okay, acknowledgement. These are the three people mostly involved in the studies that I was showing. Matthias is the one who did all the new work. Our collaborators, Rudy Eversole, Jut Bela, Paula Picotti, they are the ones that contributed to the work I was showing today and funding. And I thank you for listening.
We hold the questions over coffee. Since we are a bit out of question.
I can ask it, but it's more like sort of the relationship between the PKA signaling and just sort of the way that we do experiments in yeast, serial passaging, things like that. I don't know if you saw, but when we were mapping suppressors of various deletion alleles or TS alleles in yeast using SGA just to find linkage groups, and you could map the suppressors. And a very high percentage of the time we saw passenger mutations, which were loss of function mutations in IRA2, MEA2, anything that negatively regulates PKA signaling. And the reason we think from doing experiments is because when you have mutations in those negative regulators of PKA signaling, you do a couple more divisions before you so you just become overrepresented in the population.
Yes, yeah, they're very interesting effects. You really have to think about this twice always, because if you have these recessive mutations, they consume glucose much slower than the fast-growing cells. And then when you have them in culture, you really have to pay attention to these effects. Absolutely. Yeah.
Cool.
Okay, thanks.
Let's